Hello, my name is Eddie Polanco, and today I'm going to talk about how quantitative phase imaging can be used to help pair the right cancer patients with the right cancer therapies. I know we all know that this is what precision medicine is all about, but I want to start by discussing two important categories that were defined by Friedman et al. In 2016. The first is targeted precision medicine. This uses the genetic profile of tumor cells to identify targetable mutations for which the best therapy is already known. This is often what we are thinking about when we talk about precision medicine. However, there are two important problems with this approach. The first is that not all patients have a targetable mutation, making it impossible for them to take advantage of advances in this field. The second is even targeted therapies often result in a partial response, from which the tumor readily grows back even more aggressively than before. The second important category is functional precision medicine. This uses a functional assay, such as an in vitro model system, to identify therapies or combinations of therapies that have an effect on cancer cells derived from actual patients. These assays have strong clinical relevance and have been demonstrated to obtain results that are predictive of patient outcomes. The workflow for these functional precision medicine techniques often start with the biopsy of a patient's tumor. This is usually done to validate malignancy. However, biopsied cells can also be expanded to be used for one of these functional assays. Sometimes this expansion can take weeks or months, which is a long time when trying to obtain results for a time-sensitive disease such as cancer. Here are some examples of some of the types of functional assays that exist. As you go down this list, the model system becomes more representative of the tumor microenvironment. However, more cells are required for these types of assays, increasing the expansion time required. For example, in the murine xenograft model, it takes a long time for the tumor to grow and to perform the assay. Sometimes the time and effort required makes it difficult to test many different combinations of therapies and replicates, especially when scaling up to using these assays to help as many patients as possible. For this talk, we're going to focus on these 2D in vitro systems because they require the fewest number of cells, and maybe we can even eliminate the expansion time altogether. In general, these 2D model systems can provide results within 24 to 72 hours and require the fewest number of cells to make clinically relevant predictions. Many techniques such as cell titer glow have a demonstrated clinical relevance in this context. Cell titer glow is an endpoint assay that measures ATP levels in treated samples relative to the ATP content in the control in order to measure cell viability. This is the approach used by our collaborators in Wellm Lab at the Huntsman Cancer Institute at the University of Utah and is a gold standard technique for measuring the efficacy of cancer therapies with a high level of clinical relevance. To briefly describe our approach, we are using a 2D in vitro model system, and we are using quantitative phase imaging to measure the growth of cells in response to cancer therapies. Quantitative phase imaging measures the mass of cells by measuring the phase shift as light passes through them. Quantitative phase imaging readily measures cell mass because cell mass is directly proportional to the phase shift as light passes through the cells. Quantitative phase imaging measures growth as the change in mass over time. So by measuring mass repeatedly throughout an experiment, we can measure cell growth at all points in time during the experiment. The QPI technique that we are using is called differential phase contrast. Differential phase contrast measures the mass of individual cells using a standard compound microscope with the illumination source replaced by an LED array. The LED array allows you to quickly switch between illuminating your sample with one side of the array and then the other, which allows extra information to be captured by the objective lens from which the phase can be recovered. In this example, we show how when you illuminate from the bottom of the LED array first and then the top, the phase gradient can be computed by subtracting the two images from each other and then dividing by the sum. This process can be repeated for the orthogonal axis by illuminating with the left and right sides of the LED array. This information can then be used to compute the phase gradient along this orthogonal axis. The phase gradients together can be used to compute the phase shift at every pixel. Since the mass is directly proportional to the phase shift, we can use this information to compute the mass distribution in these cells. Here's another cell mass distribution. To compute the mass of the cell, we simply integrate over the area of the cell. To measure cell growth, we average the mass of each cell at each point in time and fit a line to this data. The rate of change or slope of this line is the average growth rate of cells in this experiment. Here you can see we use the normalized mass because the mass of a cell varies from cell to cell and based on their position in the cell cycle. Therefore, to find an average mass that is meaningful, we divide the mass of each cell at each point in time by its initial mass, so that every cell starts with a mass of 1. When we measure cell growth in this way, the units are 1 over hours, and we call this a specific growth rate because it is readily compared to other cell growth measurements, such as cell counting. An important advantage of QPI is that it resolves growth dynamics at every point in time during the experiment. In this example, you can see that the control demonstrates robust growth throughout the entire experiment. When we look at the 5 fluorouracil treated cells, we can see that the cells grow robustly throughout the beginning of the experiment, indistinguishable from the control. At around 8 to 12 hours, you can see that the 5 fluorouracil cells grow slightly slower than the control, and at around 18 hours, their growth rate starts to become negative as they start losing mass, indicating cell death. This is an important advantage of QPI because it would be impossible to see this type of response using a time-averaged assay, such as an endpoint assay. To implement QPI, we use an automated system such that the microscope travels from well to well following this snaking pattern through the plate. Our automated QPI system completes an imaging cycle every 20 minutes, measuring 9 locations per well in this 96 well plate for a total of 864 locations. With 864 locations and 10 to 20 cells per location, we image approximately 10 to 20,000 cells per experiment. To visualize this data, we compute the specific growth rate as shown on the previous slide and plot it as such so that you can readily see the specific growth rate in each well on the plate. Focusing on 2 of these drugs, we can see that a drug like fulvestrant appears to have little effect on these cells because even at the highest concentration, the growth rate appears to be affected little if at all. We can see that on the dose response curve as well, as a straight line that is approximately 1, demonstrating that at all concentrations, there's little if any effect on the cells. We would say that these cells are resistant to fulvestrant. On the other hand, we can see that a therapy like doxorubicin is highly cytotoxic because even at the middle concentrations, the growth rate is reduced by about half, and at the highest concentrations, we can see that the growth rate is dramatically affected. When we compare this to the dose response curve, the curve turns sharply downwards, indicating that the cells are highly sensitive to this drug. Looking at another experiment using MCF7 cells, we can see how another cell line is affected by the same therapies. We can see that from looking at the specific growth rates, it is difficult to tell which drugs the cells are sensitive to. However, when we look at the dose response curve, it's quite clear that these cells are also sensitive to doxorubicin as well as hydroxytamoxifen. We can also see a sensitivity to 5-FU, although to a lesser extent. An important validation step here is to compare the results from this assay to cell titer glow, since that is the gold standard technique for measuring sensitivity and resistance in cancer cell lines. When we compare these results to cell titer glow, we also see the high sensitivity to doxorubicin as well as to hydroxytamoxifen. This is very promising because this is the data we want to replicate using our system to demonstrate its clinical relevance. And we can also see this partial response to 5-FU. The way we quantify these curves is using a measure called the IC50. The IC50 is obtained by fitting a sigmoidal curve to the data and then finding the concentration at which there is 50% cell viability. When we compute the IC50 using each of these techniques, in many cases such as doxorubicin and hydroxytamoxifen, we can see that the IC50 is very similar. 5-FU in this case shows a different IC50 from each experiment, which is likely due to the fact that we did not go to high enough concentrations for the 5-FU treated cells to fall below 50% viability. By increasing the concentration of this experiment, we expect that the measured IC50 values will match more closely. Another experiment I want to discuss is using the MDA MB231 cells with a different drug panel. Again, you can see that these dose response curves are remarkably similar. Both experiments show little, if any, response to carboplatin, and they both kind of show this response, this weird response of being really sensitive at every concentration, even the lowest concentrations, and then this high sensitivity to vinblastine. we can see that when we look at the IC50s, that they're largely similar, but we can see that the IC50 for Burnapant is different between the two experiments, but the dose response curves for Burnapant look really similar. By ensuring that we use the correct range of concentrations for the cell viability to cross 50%, such as in the cases of Burnapant and docetaxel, I'm confident that we will iron out these small differences in measured IC50s between the QPI experiments and the cell titer glow experiments. Now that I've shown that there are strong similarities between the results of cell titer glow and quantitative phase imaging, I want to return to the important advantages QPI has over cell titer glow. Time-resolved growth dynamics allow us to see, for example, when a drug starts to affect cell growth. Two examples here are 5-FU and the doxorubicin. The doxorubicin, it is clear that almost immediately it starts to affect the cells and the cells start to lose mass and die. With the 5-FU, on the other hand, for the first 8 hours or so, the cells grow almost indistinguishably from the control, and then only then do they start growing at a slower rate, and then only at 18 hours do they start losing mass and dying. These insights are important because they help us plan experiments by helping us optimize our experiments to obtain the most statistically relevant results. This time-resolved data also allows us to study the effect of drugs during different time intervals, such that we can really understand the true depth of response that cells exhibit to each drug in the assay. I want to at least mention one other important advantage of QPI over the standard endpoint assays, such as cell titer glow. QPI obtains single cell data, which allows us to not only measure the population average responses to various therapies, but also the response of individual cells. This gives us the ability to investigate partial responses in ways that would be impossible using population average measurements. Here we bin cells by their specific growth rate as shown on the left side. The colors represent the fraction of cells in each bin such that the entire column adds up to 100%. Along the top, each column represents either the control or a different treatment group. When we look at cells in the control group, we can see that there are a few cells that grow kind of slowly, a few cells that grow rather quickly, but most cells kind of fall into an intermediate growth rate. Though we can see that when we treat them with different drugs, such as the 5-FU or the doxorubicin, this distribution gets shifted towards lower growth rates or even negative growth rates in the case of doxorubicin and hydroxytamoxifen. We can also see that another drug, such as lapatinib, the growth distribution is not really affected very much at all and is very reminiscent of the control. At the beginning of this talk, I mentioned how a partial response is something that is important to identify and avoid when trying to determine the best therapy for a patient. Measuring how individual cells respond to therapies is an important step towards determining whether a partial response is due to all cells being affected a little bit or whether some cells are being dramatically affected while others are completely unaffected. This is an important step in understanding why partial responses occur. In summary, QPI can be used to measure the growth rate of individual cells by measuring their change in mass over time. This allows us to measure the growth dynamics of cells at all time points, giving us more information than traditionally available using time-averaged assays. Since QPI measures the growth of individual cells, it also allows us to study the growth heterogeneity of a population of cells in response to cancer therapies, allowing us to identify and study partial responses to therapy, which becomes especially important in the clinical setting when trying to determine the best therapy for a particular patient. Future work will further demonstrate that QPI can be used to obtain very similar results to the gold standard technique cell titer glow. Furthermore, we will test this tool against patient-derived samples and compare our results to patient outcomes in order to validate the clinical relevance of this tool. I would like to thank the CDMRP and the Department of Defense for funding this work. I would also like to thank our collaborators, Dr. Phil Bernard and Whelm Lab at the Huntsman Cancer Institute at the University of Utah for their help in obtaining samples and for helping with the cell titer glow experiments. Thank you for listening to my talk, and with that, I will take any questions.